Hi, I'm here to tell you about a new research tool for scientists and microbiologists who want to understand the behaviour of microbes in various scenarios. It's called Speedy Breedy. It's designed specifically for people doing research into food or cleaning products, developing pharmaceuticals, cosmetics or healthcare products, or developing procedures for maintaining sterility in products, inventing pet food, or even looking at shelf life of healthcare products. It's also perfect for academic researchers with an interest in a whole host of areas, from medical to food to pharmaceutical. It's for anyone who needs to understand how microbes behave in a variety of conditions and media. Currently, test equipment varies from large lab-based machines that can test a couple of hundred samples to manual systems using this simple agar plate, a technique invented in 1879, over 130 years ago. No matter what you currently use, Speedy Breedy can change the way you work for good. Speedy Breedy is a clever little device. It's a microbial respirometer, an instrument that will rapidly grow and monitor bacteria and fungi in liquid. Let's take a look at how Speedy Breedy works. Speedy Breedy has two chambers. Inside each one is a disposable culture vessel. A new one is needed for each experiment. The culture vessel is sealed, allowing very sensitive pressure measurements to be made once placed inside Speedy Breedy. To run an experiment, you just introduce a growth medium of choice, perhaps containing a test material such as a growth inhibitor, into the culture vessel and then add the sample. Place the culture vessel into the culture chamber, then press the start button on Speedy Breedy. Simple. Inside the culture vessel there's a paddle mechanism which turns to keep the broth and sample optimally mixed. The paddles are magnetically driven. If microbes are present, they grow and respire, altering the pressure in the headspace of the culture vessel, which is detected by this sensor, which means that microbes are present and growing. Protocols are designed using Speedy Breedy software that is loaded onto a Windows PC. While Speedy Breedy comes with a set of standard protocols, researchers can design their own and download them to Speedy Breedy. The three variables that make up a protocol are temperature, the speed the paddles turn, and also how frequently a pressure reading is taken from Speedy Breedy. Protocols can be stored and retrieved on the PC. Once the culture chambers have been populated, Speedy Breedy starts to heat or cool and take readings to the predefined protocol. The two chambers can be used to run two experiments to the same protocol, or to compare a sample to a control. This animation shows a 500-minute experiment speeded up. Initially, the curve shows the vessel heating. Then, as time goes by, bacteria consume gases such as oxygen, or produce gases such as carbon dioxide, changing the pressure in the vessel headspace, which you can see here as the curve dips, then dramatically climbs. The dip is where bacteria have grown to large numbers and begin to modify the pressure in the headspace. So how long does it take? Well, that depends on the degree of contamination in the sample. For a common organism such as E. coli, contamination of only 50 organisms growing in TSB broth takes a little over 500 minutes for detection. Higher contamination levels will be detected much faster. Where an experiment starts with high numbers of organisms, and the measurement being made is rate of gaseous exchange and microbial activity, results can be generated almost immediately. The brilliant thing about Speedy Breedy, apart from its ease of use, is the fact that you can see what's going on inside the culture vessel. Speedy Breedy turns microbial activity into data, which means that by connecting Speedy Breedy to a PC at the start of the experiment, you can watch the warm-up curve and then see how the experiment is progressing. Speedy Breedy lets me visualize my experiment in near real time. Experimental results can be uploaded to a Windows PC by connecting Speedy Breedy via this USB port. Alternatively, data can be transferred via the memory card. So it's an incredibly useful tool, Speedy Breedy, in the lab, specifically designed to be well within the budget of even the most financially challenged researcher. www.speedybreedy.com